These are my disclosures. So I think all of us know what antibodies are. I just include this sort of basic 101 here that, every antibody has a variable region with very distinct specificity that targets a specific antigen, and it has an effector region which mediates its effectiveness. And so the effector region is what actually mediates the activity of some of the naked IgGs that we use in the clinic through complement-dependent mechanisms, ADCC, meaning activation of cellular cytotoxicity through FC receptor-mediated mechanisms, or phagocytic-mediated cell death. And I think that in many ways, at least the anti-CD38 clearly works this way, and the SLAM-F7 antibody also in some way works like this. We heard mention earlier about the water boy that brings the drug. There are also antibody-drug conjugates in clinical development in myeloma. Perhaps the BCMA ADC is seems to be moving into later stage studies. Others that have been looked at are CD138 and CD56. And one can't forget about just the antibodies that where the FC is selected not to induce ADCC or other FC-dependent cytotoxic mechanisms, but where it's the target antigen and the blockade of the target antigen that seems to be relevant. I think case in point here are the immune checkpoints where you are blocking PD-1, PD-L1, CTLA-4, and some of the immune agonist antibodies where you're actually activating the receptors. And I think the SLAM-F7 antibody, at least theoretically, also in some way works via this mechanism. So I'm going to talk mostly about, of course, anti-CD38 and SLAM-F7 targeting antibodies. I'll focus on daratumumab and elotuzumab since these are both FDA approved. So first, the elotuzumab. So this is a humanized IgG1. It has an immunostimulatory function where it can activate natural killer cells through targeting this SLAM-F7, which is a glycoprotein highly expressed on myeloma cells and natural killer cells. So it turns out that natural killer cells actually have the adapter protein EAT2 that mediates the signal through SLAM-F7 and leads to downstream signaling and granule formation, whereas plasma cells don't actually have this adapter molecule, and the antibody essentially just coats the plasma cell and is essentially a tag for recognition. And by these two theoretically synergistic mechanisms, the antibody, you would have antibody-dependent cytotoxicity mediated by NK cells. Now, what the initial phase 1 experience with this drug showed, however, is that you didn't really have very much clinically relevant ADCC going on. There was a stable disease signal in about a quarter of patients, but as a single agent drug, elotuzumab was not very interesting. It also didn't have significant toxicities other than infusion reactions typical of biologics. However, because one of the mainstays of myeloma therapy is lenalidomide, or the IMiD drugs, which can induce NK cell function and enhance NK cell function, and some preclinical data to suggest a synergy between elotuzumab and lenalidomide. Additional combination studies were pursued, and so this is just sort of a summary here of the phase 2 data for 73 patients that were treated with the combination, 2 different doses, 10 and 20 milligrams, and there was a better than expected activity for this combination, both in terms of response rate and PFS based on historical controls. And so this formed some of the rationale for this to proceed to phase 3 investigation. So this was, I think, this is the study that led to the approval of this combination of elotuzumab with lenalidomide and dexamethasone. Essentially, individuals who had relapsed following at least one prior therapy, but who were lenalidomide sensitive, were randomized to either standard dose lenalidomide and dexamethasone or lenalidomide and dexamethasone with elotuzumab, given weekly for a month and then every other week thereafter. And with the traditional endpoints of PFS and response. And what's clear was that the addition of lenalidomide to elotuzumab to lenalidomide and dexamethasone delayed progression by approximately 5 months, and led to a significant reduction in the risk of death. And this was associated with deeper responses, so greater amount of EGPR. It should be noted that this is a monoclonal antibody, and we typically measure disease burden by looking at SPEP, and so you can actually find this antibody on an SPEP. It leads to a monoclonal spike, and that's a challenge for calling somebody in CR, but depth of response seemed to be greater, and toxicity burden was not significantly increased, with the exception of infusion reactions, which occur in approximately 10% of patients, and really with the first infusion, and subsequent infusions less so. So this led to the approval of ELO in combination with lenalidomide and dexamethasone in the relapse setting of 1 to 3 prior therapies. And of course, naturally these agents are going to move into, or try to move into, the front line. So there's a host of studies which I've listed here for which readout is pending several years, looking at ELO Lendex in the high-risk smoldering population, as well as in newly diagnosed transplant ineligible and transplant eligible patient populations. So we await those studies and the challenges of comparing them to the same studies with daratumumab and figuring out what to do. So anti-CD38, so I'm going to focus mostly on daratumumab because it's FDA approved. So this is an IgG1 monoclonal antibody. CD38 is an ectoenzyme, and so the antigen specificity itself might interfere with this ectoenzyme activity that might itself lead to direct cell cytotoxicity of the plasma cell. This is also a fairly good ADC and can induce complement-dependent and antibody-dependent cellular cytotoxicity, as well as ADCP, which is the phagocytic-mediated cytotoxicity. There's also groups of cells in the microenvironment that express CD38, including suppressive T cell populations, myeloid populations, and B cell populations. And among patients who respond to single-agent daratumumab, it's been observed that some of these populations are depleted, and that there is a concomitant expansion of antigen-specific CD8 T cells. And this suggests that this drug not only targets the plasma cell, but also seems to target the microenvironment and perhaps shift toward a myeloma-specific immune response. This, of course, is a suggestion, but it's led to a variety of clinical studies to directly test this immune modulating hypothesis in combination with PD-1 pathway blockade, for example. So as a single agent, daratumumab actually does have activity, and there's in patients that are refractory to IMiDs and PIs, and essentially this was a group of patients refractory to almost everything that's available at the time when this study was run. There was still a response rate in about a third of patients, which was relatively durable, going out to approximately 2 years. What was also interesting in this group is that in the individuals where it was not apparent that you had direct anti-myeloma activity, the sort of stable disease, minor response population, there was still a pretty prolonged median OS, again suggesting this idea that perhaps there's some immune modulation and immune-mediated disease control induced by this drug, so not just anti-plasma cell-directed effect, but also microenvironment change. So this drug was FDA approved in the 3 or more prior setting as a single agent, but of course immediately was tested in combination, and so I'm just going to talk about a couple of the studies that have read out. So the first one that read out was Castor, which I thought was a very unfortunate name having come from Europe. So this study was reported first at ASCO, it was a plenary session last year. This was for patients that had relapsed after 1 prior therapy that were not refractory to proteasome inhibition, and these individuals were randomized to traditional bortezomib dexamethasone dosing or bortezomib dexamethasone with daratumumab, with sort of typical phase 3 endpoints: progression-free survival, time to progression, and response. And MRD was actually included in this analysis. I'll get to that later on. And what was clear was that at early interim analysis, this was an analysis performed with a median follow-up of a little bit over 7 months, that there was clear that there was a significant PFS difference, and so the study was halted and reported out. So the other study that read out essentially a month after this at EHA was Pollux. And this was in a combination with lenalidomide and dexamethasone in essentially the same patient population. They had to be lenalidomide sensitive, also greater than 1 or more prior therapies, standard dosing of lenalidomide, dexamethasone, weekly dexamethasone, lenalidomide for 21 of 28 days, and daratumumab along its, also according to its standard dosing program, 16 milligrams per kilogram for 8 weeks, then 8 doses every other week, and then monthly thereafter. And this study also, this was with a 13.5 months median follow-up, showed a really significant 12-month and predicted 18-month progression-free survival benefit, and was also a very positive study. And at this point, both of these, DARA is now approved in the more than 1 prior line of therapy, and so you could use this, get 2 doses of lenalidomide and show inadequate response perhaps, and try to get daratumumab in the frontline setting, I guess. But at this point, it's available in 1 to 3 prior therapies. So one of the striking things here was that there was quite a high response rate, but specifically very high depth of response with lots of CRs and VGPRs. And in this case, there is an assay that's available to distinguish between the daratumumab protein on serum immunofixation versus the monoclonal protein. and that was used in this study. The other this was really one of the first studies in the relapse setting where prospective MRD analysis was performed using next-generation sequencing approaches, and what was clear was that in both arms, the MRD rate, and I'll focus on 10 to the minus 5, because there's enough individuals achieved this in both groups to distinguish what's happening here. But there was a significant increase in MRD negative responses in the relapse setting here relative to the control in both groups. And what's kind of, I think, striking here is that it didn't really matter how you achieved MRD negativity. If you achieved MRD negativity, your outcomes were pretty darn good. The other interesting point here is if you didn't achieve MRD negativity, then but you still got DARA, you did well as well. And this again echoes that sort of minor response, stable disease observation in the single agent setting, which perhaps is related to its immunomodulatory properties, but of course that requires further study. There isn't, I think there's two relevant, adverse events with daratumumab, three probably, but the ones that are immediate are infusion-related reactions, which are pretty frequent, and it requires quite a long time, especially for first drug administration. You typically have about 6 or 8 hours of chair time, which is, I think, a major challenge in giving this drug from your chemotherapy suite perspective. These are, however, quite manageable, and we typically use Singulair, and which I think is quite important, in addition to hydrocortisone or methylprednisolone and Pepcid and Tylenol, these types of things. I think the Singulair actually adds quite a lot because it seems to be a significant leukotriene based infusion reaction, perhaps because of the types of myeloid cells that express CD38 that may be depleted with first dose or lysed with first dose. The other particular adverse event that I think is important and notable is specific to the IMiDs, which themselves cause a mild neutropenia and some hematologic toxicity, and this is somewhat enhanced by the daratumumab. Definitely manageable, but it's notable and requires attention. So there's quite a lot of excitement about this agent, and so this is also moving into the frontline setting. There is now a fully accrued phase 2 smoldering study for precursor disease, for which we're awaiting readout, and in the transplant-ineligible and transplant-eligible populations, combinations of lenalidomide dexamethasone with or without daratumumab, or sort of more, I would hazard to say, European combinations using Velcade, methylprednisolone are being investigated, as well as triplet drugs, which are really the mainstay of our frontline therapy in combination with DARA, are also being investigated. I would point out here that some of these studies are using combination of PD-1 blockade with daratumumab, also with the idea that perhaps there is this immune modulating effect, this expansion of CD8 T cells that can further be disinhibited with PD-1 pathway inhibition. And so we await to see the results of these various studies, but clearly this drug is transformational. So I mentioned one of the challenges with this medication is the long infusion time. There is also a subcutaneous formulation that's being investigated, and some of the phase 1 data was reported at ASH this year, and it was encouraging to see that both the toxicity and efficacy profile is quite similar, where the responses are seen. So hopefully this will progress, and we will soon be giving this in a subcutaneous format, which will make increase the ease of administration. So I think the question is how is this all going to get incorporated? So we've made a lot of progress in myeloma. We've moved from about 3 decades of melphalan-based therapy in low dose and in high dose, as alluded to by Sunder, to novel agents in the 2000s, and next generation novel agents, I guess. Second generation proteasome inhibition and IMiDs, now these antibodies. But I think there's now emerging immune modulatory antibodies targeting, specifically trying to affect the immune response in this disease, as well as ADCs in clinical development. I didn't talk at all about the BiTEs, of course. And so there, I think, is a lot to be encouraged about in myeloma. And I think the antibodies, although we're new to them, make us all pretty excited. And so with that, I'll say thank you very much.